storage house of the food in the animal cell, the storage house of the waste product, almost simple digestive system as a food vacuole in amoeba or living organisms, in small living organisms, just like playing a role of the kidneys in the small organisms. Like in the small organism there are no much systems are present, and that will do. So vacuole much important, but I will start from the plant cell. Vacuoles, it is a membranous organelle or the non-membranous organelle? That is a question first of all, because when I am discussing, I am much interested to know that which structures are present in the prokaryote and which structures are present in the eukaryote. Prokaryote, well-defined nucleus present, mean membranous organelles are present, mean the structures which are present in the cell, those structures are called organelles, and they are with the membrane, mean they are membranous organelles. So vacuoles, I will tell you, they consist of a membrane or not membrane. That suspense will be out when I will draw this figure. Look here, this circle will tell it consists of membrane. And vacuole membrane is so important. First of all, I will name this membrane, that is not written in the book even. You will write here tonoplast, Tonoplast, tonoplast. So tonoplast is the membrane of the vacuole. Now when you were studying the cell membrane, which important thing you studied in the cell membrane? You saw two molecules in the cell membrane. You saw significant property of the cell membrane. You saw cell membrane was living structure. You saw some molecules were going into the cell by the protein door. Some molecules were going through the lipid layer. The polar molecules were moving through the protein channels, and the non-polar molecules were going to the bilipid layer. If you will recall that lecture of the cell membrane, you will see that I told you that two different ways for two different molecules. What is the definition of the polar molecule? The charged molecules in which the partial plus or partial negative charge is present, such molecules are called the polar molecules. And the molecules which do not consist of the charge, mean they are without the charge, the partial positive and the partial negative charge will not be present. Such molecules will be called as a non-polar molecule. There is a separate way in the cell membrane. Polar molecules will enter through the protein door, mean that protein layer. Do you know that protein layer was continuous or discontinuous in the fluid mosaic model? Did you remember? Discontinuous, of course, that is a discontinuous. The discontinuous layer in the protein, discontinuous layer in the protein, and the continuous layer in the lipid layer. Continuous layer in the lipid layer, the bilipid layer, which is a continuous, that will allow the non-polar molecules to pass through it, and the polar molecules will enter through the protein gates. Mean, if you, why I am telling you cell membrane in the tonoplast, reason, this membrane also do same thing as the cell membrane. This membrane also, like cell membrane, allows some molecules to enter into the vacuole, and some molecules will not be able to enter into the vacuole. This is a much significant point. If you will see the cell membrane, tonoplast is doing the same function as cell membrane is doing. It means that is a living structure. It means that is a just door of the house. It means just you are allowing some people to enter into your house, and some people are not going to enter into your house. You are refusing. Why you are coming without my permission? Just like the tonoplast, just like the cell membrane, tonoplast do the same thing. Tonoplast allows some molecules to enter through that, and some molecules will not be able. This selectively permeable nature of tonoplast of the vacuole made the vacuole a much top ranked organelle in the cell. Because when you will be in higher sessions, if I am going to discuss these things in the future, the time will be in our hand, and we will be able to handle the situation. I will tell you that vacuole will allow such molecules due to which plants will survive. Now I will tell you size of the molecules of plant cell. This is the structure of the plant cell. If you will see the vacuole nature, if you are watching this molecule, okay, the vacuole is large in the plant, vacuole is small in the animal cell. The vacuole is the large in the plant cell, vacuole is the small in the animal cell. The reason is that the water molecules enter into the plant cell from the high concentration to the low concentration, which process diffusion. I am not going to highlight diffusion osmosis here because separately I have to discuss these discussions, but I will tell you, when the molecules flow from the high concentration to the low concentration, the process is called the diffusion. Now this vacuole in the plant is just standing with proud, with snobbish way at the center of the cell. Why it is at the center of the cell? Reason, because it can withstand the pressure of the water. Mean, if you see the figure, I will tell you, the water enter into this vacuole, the water will collide with the wall of the vacuole. I should say wall, yeah, I should say something else, I should say membrane, why? Because the membrane of the vacuole is called the tonoplast. It is not the cell wall. The cell wall is composed of cellulose. That is a dead structure. That is not allowing some molecules to enter. That is allowing all molecules to enter into the cell. So cell wall is not the structure, is not the living structure. And cell wall, you know, that consists of the two walls, primary cell wall, secondary cell wall. You know, primary cell wall composed of cellulose. You know that secondary cell wall composed of a lignin, and lignin will make the plant cell dead structure, and that will not allow sometime the molecules to enter into the cell. So that is not the selectively permeable membrane. That is the wall. The vacuole membrane is not the wall. That is the living membrane. That is living membrane, not like the cell membrane composed of bilipid layer, protein and cholesterol and glycolipid and the glycoprotein, such molecules which are attached with the cell membrane. Unlike that, but still it has some significant molecules which can select some molecules to enter, which you will learn. But here I am telling, just water enters from outside to inside by the diffusion or osmosis, which you will study. Okay, don't worry about these terms. And nucleus is pushed towards aside. You can see, I should show by another color so that that visibility must be clear. And nucleus will be at the one side, and what will happen? The nucleus will be at one side. The nucleus presence at one side will tell us that the nucleus is not present in the center in the plant cell. Who pushed the nucleus towards the side? The vacuole pushed the nucleus aside. The reason, because the vacuole has a power. Vacuole have a force due to which it is pushing the nucleus towards the one side. So vacuole push the nucleus at one side. When the vacuole push the nucleus at one side, it become little hard. It can protect the cell. The pressure develop inside the vacuole. So why the pressure develop inside the vacuole of the plant cell? The reason, the water molecule is inside. So how the water molecules are able to create the pressure inside the vacuole? I tell you, just see, this is a water molecule, one molecule. Sorry, that is not good, because figure I have to see. Just hold on, this color will finish. Okay, now you are making this red color molecule that will collide with the membrane of the vacuole. Not one molecule will collide, many molecules will collide with the membrane of the vacuole. When they will collide with the membrane of the vacuole, they will create the pressure, because the force, the collision the force per unit area is called the pressure. That will create the pressure. When they will create the pressure, such pressure, I am naming a new term for you, and you have to see this term, because I know when I will use the term first time, that will be little difficult for you when I will use 50-60 times, because I am naming all terms again and again. You are listening in today's lecture as well. That pressure is called as Turgor pressure. Turgor pressure, Turgor, Turgor pressure. Now, Turgor pressure means the water molecules will collide with the membrane of the vacuole, and the membrane will collide with the cell wall, and cell wall withstand that pressure. That will make the plant cell more hard and the protective in the nature. This thing will not happen in animal cell. So why this thing, Turgor pressure, is not present in the animal cell, it is present in the plant cell? The reason, the animals have other protective system. If you are talking about yourself, you have the bones, you have a cartilage, you have a much developed skeletal system to protect your body. There is no need of Turgor pressure. Of course, you have no cell wall as well, because the nature has given you bones. The bones are developed structures than these minor simple structures. So we'll go back to the vacuoles. Inside animal cell, if I will say that one vacuole which just behave like a complete digestive system, you did not study digestive system, you will study in the next chapters, but that system will digest the food molecules. That is the food vacuole. Exactly the same scenario I will explain after 2 or 3 lectures, in which you will see that one structure, lysosome, will combine with the food vacuole, the lysosome coming from the Golgi complex. Lysosome will consist of enzymes. You did not study lysosomes, you will study in my next lecture. I am just giving you importance of the food vacuole. The food vacuole consists of the food, the lysosomes will consist of digestive enzymes. Lysosome food vacuole will come together. You will see the figures in that session I will make, and you will see that lysosomes will give the digestive enzymes to food vacuole, and digestive enzymes will change the food into smaller components, like the protein will change into amino acid by enzyme pepsin. Similarly, the protein will change into peptones, because aminopeptidase, another enzyme, will change peptone to amino acid. You will study in digestive system, don't worry about these terms. Amylase enzyme in oral cavity, in buccal cavity, in your mouth, will change the starch to the glucose, and the lipase enzyme will change the lipid fats into the fatty acid and the glycerol in the small intestine. So what will happen? The vacuole membrane tonoplast never mean that the vacuoles are of one type as in plant cell. No, in the animal cell they are of many types, like food vacuole I am discussing, behaving like a digestive system. Question, sir, animals consist of digestive system, we consist of digestive system, esophagus, stomach, intestine. Why there is need of the food vacuole? I am not talking about your body. I'm not talking about the developed animals. I'm talking about the less developed organisms, which are just unicellular. They consist of one cell, like amoeba, paramecium, euglena, and they do not have a digestive system. They need to develop very simple digestive system, like food vacuole. So this is the food vacuole. This is not a vacuole which stores water like a plant cell and will make the turgidity, turgor pressure. It means vacuoles have different functions in the different living thing. So vacuole never mean they are doing only one function. If you see the food vacuole, what is happening? The lysosome is coming. What is lysosome? I am going to explain in the next session, don't worry. Lysosome consists of digestive enzymes. How these digestive enzymes are produced, I will discuss about the Golgi complex that I have recorded. You will see, that is upcoming sessions. Golgi complex produce the digestive enzymes. They both come together. Digestive enzymes move from the lysosomes to the vacuole. This vacuole is the food vacuole, remember. This vacuole is a food vacuole. They will digest the food. And complex molecule, what is the digestion? The complex molecule, the major molecules, the large molecules changes to small molecules by help of enzyme. When it is digested, when small molecules will be formed, that small molecules will move from the food vacuole to the cytoplasm, mean that will be distributed to all parts of the cell. Why there is a need to distribute this digested food? What is digested food? Small molecules. What is the name of the small molecule? I will say glucose only, because glucose need is much necessary in the cell and the bodies. Why glucose need is much necessary? Just recall the lesson which I taught you about the respiration in the mitochondria. The respiration is the process which will not take place. We are not living. The respiration is the process that the glucose will not be available and will not react with oxygen in the mitochondria. A chain of chemical reactions, not one reaction. A network of chemical reaction like Krebs cycle, electron transport chain. You are going to study little a bit in the respiration chapter, but detail in the second year. What will happen if respiration will not take place? Energy will not be produced. Now you are getting the idea. The food is not digested, energy will not be produced. The glucose will not be produced. The glucose will not diffuse from high concentration to low concentration outside. This glucose comes out. It goes to mitochondria. It goes to mitochondria inside liquid. You know the liquid of the mitochondria, because I discussed in the last sessions. Just recall, I am waiting. I will just feel by dream that you are telling me answer, because you are not sitting in front of me. Mitochondrial matrix, you are right, the mitochondrial matrix, of course. And the inner foldings due to which surface area was increased? Cristae. What is the membrane which was increasing the surface area and the chloroplast? Just like the cristae inside the chloroplast. Thylakoid plates. Thylakoid plates combine to form the structure called as granum. The many granum collectively called as grana. Why grana are increased surface area? Why surface area is increased in the grana or thylakoid membrane? Reason is that photosynthetic reaction will take place. Why membranes are more? Why membranes are more in mitochondria? Why membranes are more in chloroplast? Why nature increased the surface area of these membranes mean by inner foldings, by doing folding inside inner membrane of mitochondria. That are much complicated chemical reactions in the second year, first year, but I will tell you here that when I was teaching mitochondria, I remember F1 particles I have used. These F1 particles which are present in the inner membrane of the mitochondria, they produce enzymes. Enzyme chapter you will also study. Enzymes are present, enzymes will not be produced, the metabolic reactions will not take place, and glucose will not react with oxygen, and carbon dioxide water will not be produced, energy will not be produced, exothermic reaction will not take place, ATP small packets of energy will not be produced. I will not be able to speak, you will not be able to play, you will not be able to listen the lectures, you will not be able to do activities. So all living things, including plants, including animals, they are begging for respiration. Respiration is not present, we are not living. So you got the idea, food vacuole, just behaving like a very simple digestive system, very simple digestive, getting the enzymes from the lysosomes. Factory of the cell, I will tell you, because if you want to form the biscuits, you will purchase the milk from the market, you will purchase the wheat from the market, and you will process and you will make the biscuits, then you will pack in the packet, you will export to the country, and you will be export outside the country. This word you will note here, this sentence I will teach a full topic by this way. I told Golgi complex about, you will see in the upcoming session, you will understand the point I told you, making of biscuits. Because whatever the products which are required in the cell, that is the cash and carry shop of the cell. You will go to cash and carry shop, Golgi complex. Mr. Golgi complex, I need that enzyme. That Golgi complex will give you, because that is a just shop. That is a factory. That is the factory of the cell. Digestive enzymes are needed. I need pepsin. The pepsin he will make. If you need the amylase, the amylase will be given to you. So this is the lecture number. This is the session which is the